medical staff of the Kent Northwest Kidney Center with a shout out this month to our close cousins in Renton. This is the Thanksgiving edition of our monthly in-service, and today we're going to give an update on xenotransplantation, or the transplantation of organs from animals to humans. I last reviewed this topic in January of 2022, and the previous video is still available online if you're interested in knowing more about the history of this field. Today's video will instead focus on developments over the past few years, specifically with respect to pig kidney transplantation. The field is still in its infancy, but since we last discussed this topic nearly 4 years ago, we now have some clinical data to share. Xenotransplantation is a cutting-edge treatment for end-stage renal disease, but it still faces biological, ethical, and regulatory challenges. The objectives of today's talk are to understand why pigs have been chosen as the preferred source of xenotransplant kidneys, review the genetic modifications which have helped make xenotransplantation possible, and be familiar with the clinical problems involving pig kidney transplants. Let's begin by discussing pigs. Approximately 1 billion pigs are grown worldwide each year, and as an aside, the 1 billion pigs produce more carbon dioxide than the entire automobile industry every year. Most of the pigs are raised in China, about 730 million, while as the US raises about 120 to 130 million pigs per year. Pigs, of course, are used for many purposes: food, fertilizer, leather and collagen products, fat and lard used in cooking, cosmetics, or various industries, pets, biotechnology. Most of the artificial heart valves that we use are made of pig valves. Heparin also comes from pig intestines. Biomedical research involves pigs in studying cardiovascular disease, diabetes, and obesity. And xenotransplantation. Xenotransplantation is the process of transplanting cells, tissues, and organs from one species into another. In the medical world, it specifically refers to transplanting animal organs into humans. As you all know, organ shortages are severe. There are thousands of patients who die every year while waiting human organ transplants. There are currently about 520,000 people on dialysis in the United States, and there are about 110,000 people on the kidney transplant waiting list. Xenotransplantation has the attractive potential to provide a much larger supply of organs than we currently have. There are many reasons why pigs are the preferred animal source for xenotransplantation. There are fewer ethical and cultural concerns with pigs than there are with primates, say chimpanzees. Pigs are anatomically and physiologically similar to humans. They can be bred in controlled, disease-free environments. Organs are close in size to human organs. Pigs grow quickly. You can take a pig from 0 to 6 months, at which time it's full grown. And they are, of course, easy to breed. And genetics can be edited to reduce immune rejection. This is an important part of the story of pig kidneys and transplantation. Unmodified pig organs are immediately attacked by the human immune system due to molecular incompatibilities and risk of something called hyperacute rejection. But these days, researchers can use gene editing tools such as CRISPR to remove some pig genes and add human immune compatible genes. So how exactly are pigs genetically modified for xenotransplantation? So in general, researchers try to knock out pig genes that trigger immune attacks on the part of a human host. They insert human genes that help regulate blood clotting, inflammation, or the immune response. And they try to minimize the risk of infection from something called porcine endogenous retroviruses via gene editing so they don't pass on to a human host who receives the kidney transplant. Okay, so what's involved specifically in removing pig genes that trigger human immune attack? So these are so-called knockouts. So the pig genome naturally contains molecules that the human immune system sees as foreign. There are 3 particular genes that can cause what we call hyperacute rejection within minutes of transplantation. One is something called alpha-gal, which is this GGTA1 knockout. Alpha-gal sugars on pig cells cause a massive human antibody attack. And delete it reduces the chance of immediate rejection. There's something called a CMAH knockout. Again, CMAH is a sugar on the surface of pig cells to which humans strongly react. And then there's something called the B4GALNT2 knockout, which eliminates something called the SD antigen, another immune target. And so when you knock out all three, we call this a triple knockout. And so essentially all pig kidney transplants used are triple knockouts. Next, researchers add human genes into the pig genome to make pig kidneys more compatible, and these are called transgenes. So they insert selected human genes that help regulate immune reactions, blood clotting, inflammation, and these make the pig kidney behave more like a human organ in key biologic processes, and helps the pig kidney survive longer in a human circulatory system. So there are a whole bunch of added genes, and here are some examples, something called immune regulating genes, membrane cofactor protein, along with the decay accelerating factor. These mitigate potential damage by something called the complement system, which is part of our immune system. You have these white blood cells called macrophages, which would attack the kidney, but you can insert this signal, this HCD47 signal that tones down their response. Basically, coagulation inflammatory genes, and also genes involved in the human clotting pathways to reduce the chance of clot formation and inflammation. Finally, hemoxygenase 1 protects against stress and inflammation. The pig genome is also modified to reduce the chance of viral transmission. So virtually all pigs carry something called the porcine endogenous retrovirus, which is embedded in their genome, it's just part of their DNA. And when pig organs are transplanted to humans, particularly those given immunosuppression, there is a theoretical risk that these porcine endogenous retroviruses could infect human cells. And again, it's part of their DNA, and so it's present in all pig tissue, including their kidney organs. So the researchers basically use CRISPR to inactivate large sequences of these porcine endogenous retroviruses, basically to remove them from the genome. And the importance of this is that it can reduce the chance of a cross-species viral transmission. Pig kidneys can grow after transplantation in response to human hormones, and they can get up to twice the size of normal human kidneys. And so it's important to insert certain gene edits that prevent the pig organ from growing too large after it's transplanted. Researchers also have made attempts to modify what are called HLA genes. So HLA genes are genes that produce proteins that sit on the surface of human cells. They help the immune system distinguish itself from foreign proteins or non-self. In the world of human organ transplantation, there are 6 important HLA proteins or antigens, and they usually refer to class HLA-A, HLA-B, or HLA-DR antigens, which are very important for matching donors and recipients. So each person has 2 of each, 2 HLA, 2 HLB, and 2 HLA-DR, one from each parent, and so there are 3 types times 2, or 6 antigens. So a 6 antigen matched kidney is also colloquially known as a perfect match kidney, and that minimizes the risk of a recipient's immune system rejecting the organ. Now, when a human receives an organ from like a pig, which contain foreign HLA molecules, the immune system will mount a very strong rejection response. And pigs have their own versions of HLA antigens. They're called swine leukocyte antigens, which can trigger hyperacute or acute immune rejections in human recipients. So what researchers have done is to either knock out the SLA class 1 or 2 molecules in pigs so they don't activate T cells in humans, or introduce human HLA molecules into the pig to provide cell signals to the host's natural killer and T cells so that rejection is minimized. So how do researchers make these edits anyway? Well, there are different forms of technology available, including something called CRISPR, or somatic cell nuclear transfer, where you basically clone an edited cell to produce a whole animal. There are many different gene edits that have been done to pigs that are bred for xenotransplantation. One company, for example, boasts having made 69 genomic edits, so 69 changes of the pig DNA in order to inactivate porcine endogenous retroviruses and add various human genes and delete certain pig antigens. Other companies have focused on fewer gene edits, more with the goal of preventing rejection on the part of the human host. In general, the more edits that are made to the pig genome, the more complex breeding and production become, but more edits also can make organs more human friendly. And as gene editing tools such as CRISPR get better, doing more edits is more feasible. Let's now review the available clinical cases involving pig xenotransplantation. To date, there are 4 pig-to-human kidney transplants that have been done so far. These transplants have mostly been done under FDA expanded access or compassionate use protocols, but not as part of a full, large-scale clinical trial. These are pioneering cases. The field is still very experimental and long-term outcomes remain uncertain, but I think it's worthy to mention the stories and names of the individual patients who have been willing to receive an experimental transplant and recognize their contributions towards the benefit of the broader medical community. Rick Slayman was a 62-year-old man from Weymouth, Massachusetts, who had end-stage renal disease secondary to type 2 diabetes mellitus and hypertension. He previously had received a human kidney transplant in 2018, but it had failed. He was the first person ever to receive a genetically modified pig kidney in a living human. He received the transplant on March 16, 2024. The pig kidney had 69 genomic edits. He died on May 11, 2024, about 2 months after the transplant. The hospital said his death was due to a sudden cardiac event. It is not clear that the pig kidney was rejected by his immune system. Lisa Pisano was a 54-year-old woman from New Jersey who had both heart failure and end-stage kidney disease, so she was very ill. She received a genetically modified pig kidney and a heart pump called a left ventricular assist device. The pig kidney was removed about 1 month later in May of 2024 because of complications related to other health issues, and she later died in July of 2024. Tawana Looney is a 53-year-old woman from Alabama. She generously donated one of her kidneys to her mother in 1999. Her remaining kidney failed due to complications from pregnancy, something called preeclampsia, which features very high blood pressure. She was also considered highly sensitized, that is, her immune system would likely reject a human kidney. So she received a pig kidney that had 10 gene edits on November 25th, 2024 in New York. Unfortunately, she developed an infection in the postoperative period that required a reduction in immunosuppression, and that led to immunologic rejection of her kidney, which lasted about 130 days before it had to be removed because of acute rejection, after which she returned to dialysis. And finally, Tim Andrews, a 66-year-old man from Concord, New Hampshire, who'd been on dialysis for 2 years. He also had heart disease, had a heart attack in July of 2023. He received a pig kidney with 69 genomic edits, so the same kind as the first transplant that we talked about. On January 25th, 2025, the kidney lasted 271 days but was removed on October 23rd, 2025 because of declining function, after which hemodialysis was resumed. Because of these remarkable and heroic volunteers, the Food and Drug Administration has given clearance as of February 2025 to 2 biotech companies, United Therapeutics and Genesis, to start clinical xenotransplantation trials. United Therapeutics plans to begin with 6 patients, and if experiments go well, expand to up to 50. So why do pig kidney transplants fail anyway? There are several reasons. The first is immune rejection. So this can still occur despite genetic modifications. Acute rejection occurs when T cells recognize pig proteins or antigens as foreign. And there's something called antibody-mediated rejection, where humans have preformed antibodies or develop antibodies after the transplant against pig antigens that may not be eliminated by gene edits. And this can occur even with triple knockout and added human transgenes because of other immune targets. There are also some problems with something called incomplete physiologic compatibility. So pig blood vessels are not perfectly matched to humans. Even with humanized genes such as thrombomodulin, clotting issues may arise. Pig kidneys may grow differently, respond differently to human hormones, and there's also a metabolic mismatch. Pig kidneys process electrolytes and waste products slightly differently, which can stress the organ. For example, pig kidneys tend to absorb more sodium and have reduced capacity for potassium excretion. Of course, the use of immunosuppression leads to increased risk of infection. These patients receive a lot of immunosuppression to prevent rejection of the kidney transplant. That can lead to infection. So, for example, Mrs. Looney's kidney began to fail after a bacterial infection required a reduction in immunosuppression. Additionally, there's the potential for opportunistic infections or infections that don't normally cause clinical disease in humans but can do so when someone is on immunosuppression. The patient's overall health and the complexity of their medical problems can also affect clinical outcomes. For example, the 4 people who received pig kidney transplants under a compassionate use protocol were not candidates for conventional human kidney transplants. And so if you have a lot of other health problems, particularly heart problems, this is going to shorten your chance of survival. Technical or surgical factors can increase the risk of xenotransplantation failure. It's more complicated than a human transplant. There are differences in blood vessel size and pressure. For example, pigs have much lower blood pressure than humans do. You need to make adjustments for pig kidney anatomy. You need to ensure proper perfusion or blood supply to the organ as well as drainage through the ureter. Part 5, where do genetically modified pigs come from? There are several companies that have invested heavily in developing genetically modified pigs. For example, eGenesis has done very extensive editing of the genome. Others focus on a more minimal but functional set of genetic modifications. Some are explicitly focused on kidneys, while others are developing other organs as well, such as liver and heart for transplant. eGenesis is one of the leading companies that's developing pig organs for human transplant. They use CRISPR and cell cloning to create their gene-edited pigs. Some of their pigs have up to 69 gene edits. Revivicor is a biotech company owned by United Therapeutics, has developed pigs with 10 gene edits. They're also working on pig hearts in addition to kidneys. Recombinetics is another bioengineering company that works on genetically engineered animals, and they've been working on pigs for organ research. Other companies called X-Transplant works on genetically modified pigs for xenotransplantation and has some pig to non-human primate experiments ongoing. So where are these special pigs raised? Genetically modified pigs are not raised on a normal farm, but instead in labs or facilities that have certain criteria that we'll talk about. For example, they are known as designated pathogen-free facilities. So these are specialized, highly controlled buildings designed to house animals or biological materials in an environment free from certain infections or pathogens. They are modern, they're well maintained, they have very secure infrastructure, they have controlled access, all of which support strict biosecurity standards, and they're used for research, breeding, and production where pathogen-free conditions are essential. There's strict biosecurity and air filtration in these facilities. The workers enter pig zones through airlocks. They have to take showers. They change into special clean suits and they wear masks. The air entering the pig area is highly filtered, as is the feed and the water for the pig is sterilized or disinfected. These measures are meant to minimize any risk of pig infections that could be passed to human recipients or infections that the humans could potentially give to the pigs. Pigs are born in a birthing center, and according to reports, these piglets are hand-raised and hand-fed to better control their health. Once they're weaned, they are moved into super clean pens where everything in their environment is tightly controlled. Despite the sterile contracts, they try to make it pleasant for the pigs. They give them toys and interact with them and make sure that they're well cared for. These pigs regularly undergo health inspection, and in particular, they're regularly screened for viruses, the 2 most notable being porcine cytomegalovirus and the porcine endogenous retrovirus. There are very strict biosecurity and regulatory standards, as well as international standards that design how these facilities are built and the donor pigs are maintained. So why do these labs matter? Well, one, safety for transplants. If you raise pigs in a really clean, controlled environment, you reduce the risk of a pig infection getting passed on to the human transplant recipient. Two, you have consistency. So these are genetically identical clone pigs, mean much more predictable organ quality. There's regulatory compliance to satisfy the FDA and other regulatory agencies, and also scalability. So these facilities are designed not just for research, but for eventual clinical production. For example, Revivicor has built a 77,000 square foot facility that could produce many organ pigs per year. There are several political and social issues that need to be addressed before pig kidneys enter prime time. Pig kidney transplantation is not just a medical issue, it is deeply entwined with politics, ethics, and regulation. There are ethical and safety considerations. There's a risk of cross species viral transmission, questions about animal welfare, genetic modification, and the need for strict regulation and oversight. Currently, the Food and Drug Administration regulates xenotransplantation, and it's very important for it to balance safety versus innovation. We all want to proceed with xenotransplantation. We have to make sure it's safe. For example, that we don't introduce a pig virus into the human population. Lots of political pressure from various groups, and there's also global variation. So different countries have different standards for what is acceptable for animal to human transplants, and so it's quite possible that other countries may allow it to occur earlier, but that doesn't necessarily guarantee safety. Ethical concerns come from animal rights groups. Critics argue that genetically engineering pigs for organ harvesting can raise animal welfare concerns, and some political groups would like to ban the use of animal transplantation, organ transplantation into humans. Also, some religious and cultural issues. Some religions forbid the use of pigs, which could influence policy decisions or inform consent protocols. And lawmakers are always under pressure to allow for religious accommodation. These organs are likely going to be extremely expensive, so who gets them? Are they going to be available to lower income patients? Will Medicare fund it? Will private insurers fund it? And so there have to be a lot of policy decisions that are involved in actually who benefits from these advances. There's certainly potential liability if xenotransplantation goes wrong, particularly if it involves the introduction of a pig virus into the general population. So governments had to be very careful in approving xenotransplantation until we have robust safety data. Because if they approve something too early and it leads to another outbreak or pandemic, then there could be a lot of problems for global health. There's also global competition to see who can first develop xenotransplantation. Countries like the US, China, and UK are racing to be the first. This can lead to political tension, but there's also a lot of pressure on the government to approve xenotransplantation so that we're not lagging behind other countries. Public perception is also important. High-profile cases can trigger political debates about ethics, safety, and funding priorities. Negative outcomes could be a nail in the coffin for xenotransplantation in this country. And there are a lot of lobbying groups that have strong opinions about xenotransplantation, as I discussed earlier. All right, let's summarize what we've talked about today. Pigs are used for all sorts of purposes for humans, but the most recent application is xenotransplantation. Pigs are the preferred animal for xenotransplantation because of their anatomy, kidney size, rapid growth and breeding, and potential for gene editing. These pigs are genetically modified to reduce the risk of rejection as well as infection. To date, 4 pig-to-human kidney transplants have been performed, all in the United States. All have eventually failed because of infection or rejection. We are deeply indebted to the pioneer patients who have volunteered for pig kidney transplants. Genetically modified pigs are grown in specialized facilities to minimize potential for infection. Pigs receive high-level security and care in these special farms, and still there's potential concern with xenotransplantation about the introduction of infection from pigs to humans, as well as health access and animal welfare. All right, let's see how much you've learned. Question number 1, why are pigs the preferred animal for xenotransplantation? Several reasons. Ethical and cultural concerns are generally lower with pigs than with primates. There's a lot of anatomic and physiologic similarity to pigs and humans. Pigs can be bred in controlled disease-free environments. Organs are close in size to human organs. They can grow quickly and are easy to breed. And finally, genetics can be edited to reduce immune rejection. Question number two, why is gene editing required in xenotransplantation? That's right, to decrease the risk of rejection and blood clotting, and also reduce the risk of infection from porcine endogenous retroviruses. Question number three, why do pig kidney transplants fail? That's right, so immune rejection, physiologic incompatibilities, for example, that can lead to blood clotting, infection, other health factors, say someone has bad heart disease, bad diabetes, etc. And finally, surgical technique challenges associated with um, inserting a pig kidney rather than a human kidney. Question number four, name one infection that humans could potentially acquire from pigs. That's right, so there's the porcine endogenous retrovirus. There's also something called the porcine cytomegalovirus. Final question, what are some ethical issues associated with xenotransplantation? That's correct. So religious accommodation for those who don't use pork products, animal rights, and availability due to high cost. This concludes the Thanksgiving edition of the monthly in-service of Northwest Kidney Center. In particular, because it's Thanksgiving, I want to use this as an opportunity to thank all of you for taking such wonderful care of our patients, as well as for your attention to these videos. This is Andy Borgenbro signing out until next month. Have a great Thanksgiving.